I have a distinct honor tonight for introducing our speaker. When we were working on this symposium many months ago, and we knew we were going to have a dinner, as we usually do, to get together after 2 hard days, have some wine, the bar is still open, break bread together. It's always nice to hear a different kind of talk. And one of the most eloquent speakers and thoughtful presenters I know of is Dr. Larry Steinman, who joins us from Stanford. And I first met Dr. Steinman when I was a resident. I'm not even sure he remembers this. He was giving grand rounds at Johns Hopkins, and I was going into neuroimmunology, and I had been sucked in by Doug Kerr to doing transverse myelitis and related diseases, and went up and introduced myself, and we had a pleasant chat. And then over the years, our paths crossed more than once, and I've found him to be an incredibly intelligent researcher, a wonderful example of how you carry on translational research between the bench and the bedside, and more importantly, somebody who never stops advocating from the patient's perspective and tries to really balance the role of research with what it's supposed to do, and that is improve the lives of the patients that we are here to serve. And tonight, I tasked Dr. Steinman with an interesting one, and that is to present his thoughts on what can rare diseases teach the more common diseases. So throughout this weekend, through all the presentations, for those of us in the room who have transverse myelitis, neuromyelitis optica, ADEM, and optic neuritis, we saw a lot of presentations where there were slides and data presented about multiple sclerosis. And we don't all have multiple sclerosis. In fact, sometimes it's frustrating to see the world of MS research moving at a certain pace where the research in transverse myelitis and NMO goes slower because there are fewer of us. Dr. Steinman's here tonight, and I'm sure will do an outstanding job to not only enlighten us, but the rest of the world on what can everybody else learn from us, and why should investments be made in the research around these rare diseases. So without further delay, I'll welcome Dr. Steinman to the podium.
Thank you very much, Ben. It's a pleasure to be here tonight, and it's a challenge to address an audience that has scientific experts and people who are even more concerned about what I'm going to say than the scientific experts, people and families who have been impacted by these problems that I talk about. So I'm going to aim the talk at a level that is about the science, but I'm going to try to explain things in a way that will be at least hopefully digestible as you're eating for all of you. And since most of you have been sitting now for 2 days listening to lectures, and then at the end of the lecture somebody says, now we'll take a few minutes of questions, I plan to talk during salad and the main course. I know there's some people who are going to be speaking during dessert, but I thought it might be possible, if somebody has a question, to even interrupt, or the talk is broken up into a few parts, and fire away with a question, because it might be very interesting to hear what the question is and to bounce an answer back. So I'll encourage that. I know we didn't bring microphones to chase people around, so when you ask the question, I'll restate it from here with the microphone. And sometimes if the question is something that I don't even know, I'll restate it in a way that it will sound like I do know it, because that's part of the beauty of that technique of restating the question, is you bring it back to the sweet spot of your own knowledge. No, I won't really do that, but it occurred to me. So the title of my talk is how rare neurologic disorders teach us about their more common relatives. So since some of the people who are buying us dinner tonight are the National Multiple Sclerosis Society and Biogen IDEC and Teva, among others, who make their lives around MS, I think that it's going to be a real opportunity to really say how we can instruct from learning about these rare diseases. In this case, I'm talking about the demyelinating diseases, but I'll get into myasthenia at the very end of my talk, how they've actually given us some incredible insights about the more popular disease. Popular in terms of affecting more people. No disease should really actually have the adjective popular attached to it, because in the English language there's sort of a good connotation, and diseases are not good. So one of the take-home messages here is that demyelinating disease, and I'm talking about MS on one end of the spectrum, and NMO and ADEM and transverse myelitis on the other end of the spectrum, might be stratified along two major pathways, and I'll explain what these pathways are because it's jargon to immunologists. One is called Th1 and one is called Th17. And that's a very popular dichotomy among immunologists. And it turns out that if we look at NMO and we look at relapsing remitting MS, that NMO is going to turn out to represent the Th17 version of demyelinating disease, whereas in relapsing remitting MS, about 70 to 80% of the time it's the Th1 form of demyelinating disease. And a lot of the differences in pathology and in the clinical course might be explainable because of this dichotomy. So it has implications for pathogenesis and opens up some really interesting opportunities for therapy. So a lot of the work I'm talking about today was funded by the Guthrie-Jackson Charitable Foundation, and we have representatives here from the Guthrie-Jackson Foundation. And that very attractive logo on the bottom about neuromyelitis optica, we describe neuromyelitis optica as the little disease that could teach us a lot about the more common disease MS. So the talk's divided into a few parts. I'm going to talk about neuromyelitis optica. The number 17 will come up again and again. It's one of the, interleukin 17 is one of the 35 or so cytokines. Those are messengers produced by the immune system that have the properties of either turning the immune system on, as some of the cytokines do, like IL-17 and gamma interferon. And there are other cytokines that turn the immune system off. And we'll hear about some of those today. So they're the chemical messengers that turn the dial of the immune system either up or down. And I'll talk about some techniques that we use to further understand what I'm talking about. We actually look at the proteins in the blood and in the brain and see if some of these chemical messengers are actually there or not. One thing you won't hear about tonight is fancy gene profiling and gene polymorphisms. That's a very important area of research, but in this talk we're going to stick mainly to the proteins and the actual chemicals, not the genes that are in play in these diseases in humans. I'll have a few stories to tell about mice, only because they taught us something very important about what to look for in humans. But I won't tell you anything about mice that is only there because I don't really care about curing experimental animals of autoimmune diseases. I'm interested in the human disease. And that is exactly the final third of the talk, some of the new therapies that we might be able to use to shut down the inflammatory part of these diseases so that the people doing the elegant work on stem cells can go in there and fix what was damaged by the immune system. So the first topic I'm going to talk about again is this chemical called IL-17, and it comes in 5 forms, A, B, C, D, E, and F, and the neutrophil. So the neutrophil has a lot of different names. It's the polymorphonuclear leukocyte. It's one of our white blood cells, and it's one of the first cells that's called into action when we get a common infection, a viral infection, a bacterial infection. The nurses and the physicians in the room, you have probably heard them say that somebody's white count is elevated, and one of the big components of the white count are the polys. So IL-17, A, B, C, D, E, and F versions of it, and there's a picture of IL-17F, actually induces polymorphonuclear leukocytes. And that is a very interesting footprint that we see in NMO. And the polymorphonuclear leukocyte, also called the neutrophil, it's called polymorphonuclear because it has a multi-lobulated nucleus. But the cell is full of explosive chemicals that are there to attack viruses and bacteria and kill them. They're called proteases, and they're there to mess up and destroy microbes. But when they get called into tissue, like the spinal cord or optic nerve or brain, and there aren't any microbes to kill, it's called sterile inflammation. And it's sterile because there are no microbes, but the inflammation is really a scorched earth attack against the brain, spinal cord, optic nerve, caused by these terrifically powerful proteins, and I'll talk about one of them, a molecule called elastase, which plays a big role in a lot of diseases, including emphysema and some of the chronic lung diseases. So a lot of the work that I'll discuss now was done by a fellow in my lab who came from, he's Canadian, and he came to Stanford to study neuroimmunology after he had started some of this work at the University of Alabama. And he's a fellow of the MS Society, but the funds that were provided for us to do a lot of this work on NMO came from the Guthrie-Jackson Foundation, not from the MS Society. So he was the first author on a paper that came out in Nature Medicine in April, and instead of just showing a lot of names at the end of the talk, I like to show the pictures of people who actually did the work. So there's Bob again, and the critical colleagues here. Brigitte de Jong is a neurologist now in Nijmegen in the Netherlands. She was a postdoctoral fellow who came from Amsterdam, where her boss was Chris Pullman, and Bob's former boss is Chanda Raman from University of Alabama, Birmingham. And the fancy title of the paper is T helper 1 and T helper 17. There's that number 17 because it stands for the pathway in the immune system that culminates in the production of this cytokine called IL-17. They determine the efficacy of interferon beta in MS and experimental encephalomyelitis. So you actually heard Ben say, you hear people that are going to get up all the time and they're going to talk about the animal model of MS called EAE, and they're going to talk about MS. And here we're at the Rare Disease Symposium. So have I now violated exactly what Ben said wasn't going to happen? No. One of the cornerstones of this paper is the fact that I'm speaking with a certain amount of confidence in my voice because of the underlying fact of what I'm going to show you occurs in neuromyelitis optica. It gives me the confidence, it's the foundation for what's the message is for multiple sclerosis. So don't get dismayed, it will all fit together. So when a paper comes out in these journals, sometimes they write an editorial, and the editorial gives people a chance from the cartoon to then really get into their salad, and if you have a glass of wine or a beer, to
start sipping it while I run very quickly through slides that have data on them. But the cartoon is more important. So they called the editorial Molecular Oracles for Multiple Sclerosis Therapy. There it goes again, multiple sclerosis. But this would be a blood test that will tell you, it's not a gene test, it doesn't involve anything all that fancy. You get your blood drawn, and it will tell you whether or not you may benefit from beta interferon if you have relapsing remitting MS, or whether or not you won't benefit. And again, there's a great connection, which I'll get into shortly, about NMO. So basically, the world was divided up into responders and non-responders. And we did this research without knowing who was who. Chris Pullman and Brigitte gave us the samples from Amsterdam, but in a coded fashion. But an MS responder is somebody who had their blood drawn, we made the observation, and then they were followed for 2 years, and if they didn't have any relapses, we call that a responder. I think that's a fair definition. And if they did have relapses, we called them a non-responder. So the non-responders have high levels of IL-17F and also high levels of beta interferon itself. Beta interferon is another molecule that's produced in response to infection, which has a very interesting way of controlling the immune response. It makes people with relapsing remitting MS better if they're not having high levels of IL-17F. But beta interferon, if you give it to somebody with neuromyelitis optica, it either has no effect or there's pretty good evidence that it actually makes the disease worse. And that's the foundation that I'll show you that gives us a tremendous insight into some of the immune biology of neuromyelitis optica. And then the lower part of the slide was actually how the work got started. We went to this animal model, which is supposed to be a model of multiple sclerosis. Well, it's not actually a model of multiple sclerosis at all. The model is called experimental autoimmune encephalomyelitis, and it had its diamond anniversary. 75 is diamond, right? Diamond anniversary in 2008. So the first paper on acute disseminated encephalomyelitis on EAE came out in 1933 from the Rockefeller Institute, and a researcher named Thomas Rivers was very interested in why individuals who were getting vaccinated against rabies were developing acute disseminated encephalomyelitis. And he was trying to set up an animal model, and he was immunizing monkeys repeatedly, and they were getting what he described as experimental allergic encephalomyelitis. So people give it a lot of attributions. Some people say it's a model for MS. It's actually was discovered and developed as a model for acute disseminated encephalomyelitis. And it has many different forms. It's not a single model. You can get it in a variety of animals, from primates to zebrafish, and it can serve as a model for neuromyelitis optica, transverse myelitis, ADEM. There's variations that reflect Guillain-Barré syndrome and chronic inflammatory demyelinating neuropathy. But shortly I'll show you an experiment that shined all the spotlight on these two pathways, the Th1 pathway and that Th17 pathway. So to make the story short, if we made EAE with IL-17 producing immune cells and gave them beta interferon, they got worse. If we made EAE paralyzed experimental animals with cells that produced not IL-17 but gamma interferon, then they got better. And this polarity in the experiment was something that, as an experimentalist, is something that you can really then use as a tremendous handhold to translate and see well, is that dichotomy, beta interferon making IL-17 demyelination worse? Is that something that actually happens in man? And we found out that it does. Whereas if you give beta interferon to that mouse that was making the chemicals from the other pathway, they got better. So here's the pathway in all of its glory. There's various lineages of white blood cells. These are the lymphocytes. They're not the polymorphonuclear leukocytes or those neutrophils. So we have the Th1 pathway, and its hallmark cytokine is gamma interferon. We have the Th2 pathway. Won't go into that tonight. We have the Th17 pathway. Its hallmark cytokine is IL-17. And then we have some regulatory cells, which I won't go into tonight. And immunologists get really worked up about these pathways. I can't begin to tell you, even in the face of contradictory data, this is what this particular article was about. It took immunologists about 20 years to get off their butts and realize that there were more than just Th1 and Th2 cells. There was, the ship was hit by an iceberg, but it took it about 10 years to sink because people said, oh, there's water below the deck. Isn't that interesting? And then, you know, finally when the ship went under, people came to realize that there were at least 3 major lineages. There are probably more, but these lineages actually do have a lot of veracity because of what some of the things I'll be able to show you. So we've been very interested in studying tissues actually from specimens that we're fortunate enough to get from our colleagues in neuropathology, and we've done all sorts of profiling of the lesion material itself. And we've used robots to sequence DNA, and we've used gene chips, and we've used proteomics. And I'll show you the protein studies. But in one of the very first papers where the gene chip technology was used, and that's what that slide shows, we take either biopsy or post-mortem material, and we extract the DNA or the RNA or the proteins, and we use one of these high-tech systems. So in the very first paper, we were writing in the very first sentence of the abstract that multiple sclerosis lesions at autopsy revealed increased transcripts of genes encoding the following: IL-6, IL-17, and gamma interferon. So we noted right from the beginning that gamma interferon, the main representative of Th1, and IL-17, which there wasn't that Th17 pathway even known in 2002, they were there. It was like discovering, if you will, a new continent, but you had no idea when you first saw it. It was just a little island that was in your vision. But now the story is getting richer. So here's another diagram. We're going to focus on this difference between what happens with beta interferon in the Th1 versus Th17, and we're going to get to this matter of the granulocyte, also known as neutrophil, also known as polymorphonuclear leukocyte. I think I'm going to try to stick to the term granulocyte, but you'll see neutrophil come up, so they're all the same thing to me. There may be hematologists who will tell me that they have different nuances, but as far as I can recall from my medical training, they're one and the same. So beta interferon makes Th17 worse and makes Th1 better. So this was the EAE experiment. We have the clinical score on one axis. 1 is very weak paralysis, 5 is a dead experimental animal, and we could induce these types of experimental demyelinating disease by giving cells that either attack your myelin and make gamma interferon or attack your myelin and make IL-17, and after about 20 days, the animals get sick after we transfer the lymphocytes. And then if we gave beta interferon to these Th1 animals, they got better. We gave it
we could give it at the time of paralysis. In this particular experiment, we gave it before they were paralyzed. It's not my favorite way of doing it. I like to give it only after the animals are sick. It worked that way too. But in the Th17, there's a night and day difference. Better in the Th1, worse in the Th17. So then we wanted to see, was that true in humans? So we got the specimens from Chris Pullman, and we learned later that 12 of them were responders and 14 were non-responders, and they were pretty well matched for a lot of different characteristics. You can see the relapse rate in the responder 2 years after the start of beta interferon was 0, and the boundaries were 0 and 0. So no one had a relapse in the responder, so that's the definition. So then it turned out that the people who were non-responders had very high levels of two markers, because we looked at about 56 markers in their blood. They had high levels. You can see over there on the panel on your right, those dots were individuals, high levels of IL-17F. You were a non-responder to beta interferon. And those people also had high levels of beta interferon before they actually started getting beta interferon. So it was almost if, had we known that they had such high levels of beta interferon, giving them more beta interferon wasn't going to help. And having high levels of IL-17F, as I'll show you, wasn't going to help. And we measured it by a variety of different platforms, because if you're going to make these kinds of claims, you better be able to back it up with more than one measurement. So that's, we did it with an ELISA, and we did it with these multiplex systems. So that was fine, and all this work was done in 2009 and published in 2010. But look at this. Brain, 2005, group from Japan showed that there were higher levels of IL-17 in the spinal fluid in neuromyelitis optica, something not seen in MS. So, oh my gosh, that's very interesting. And then paper after paper after paper says that NMO patients do not respond to interferon beta. I was told to tone it down and not to say that people get worse, but I'm beginning to think that actually they are getting worse. And the titles lay anyway what I'm saying. The first paper there, interferon beta 1 exacerbates MS with severe optic nerve and spinal cord demyelination. Paper number 2, development of extensive brain lesions following interferon beta therapy and relapsing neuromyelitis optica and longitudinally extensive myelitis. Paper number 3, early relapse. More papers coming out in the recent literature. This one, interferon beta may severely exacerbate Japanese optic spinal MS and neuromyelitis optica spectrum. A paper from Angela Vincent looking at aquaporin 4 antibodies that came out in Archives of Neurology a month ago. Interferon beta treatment in NMO, increase in relapses and aquaporin 4 antibody titers. So one of the main targets of the immune attack, the antibodies are going up when you get beta interferon. And anyway, what I was referring to a few months ago is the canary in the mine. It's more like the elephant in the room. Beta interferon is bad for NMO. It's not just a quirk, well, we'll use a different medicine. It's teaching us something phenomenally important, because beta interferon is more than just a drug produced by Biogen and Bayer and Merck Serono. It's one of the body's natural ways of responding to microbes, and has a very, very interesting role in killing viruses, but also in modulating the immune response. If you have a Th1 response going, that beta interferon is doing a wonderful physiological homeostatic function. It's one, helping to kill the microbe if it's a virus that triggered the response, and number 2, it's attenuating the response so it goes away. Because the thing about killing microbes, you want to kill them and then shut down the immune response, and that's something that doesn't happen in autoimmune conditions. So that was one of the big insights. And then we started collecting data, and I'll show you these data in different ways, but basically we saw that that same version of IL-17, namely IL-17F, was elevated in the blood of people with NMO. And then a few other chemicals were elevated that are markers for two cells. One, the granulocyte, chemicals called IL-8, another interleukin, and another chemical called GRO-A. And another cytokine drives yet another type of cell that I'm not going to talk about today called the eosinophil. And it was the exact same thing that we were seeing in the Th17 form of EAE. So this is, for the cognoscenti, not that important. Over dinner, why do you see more 17F than 17A? But it's going to be important because one of the implications here is, well, if IL-17 is produced in NMO and we have antibodies to IL-17, why don't we just use that as a therapy for neuromyelitis optica? So if we're going to think about things like that, we'd want to know, would we want an antibody to IL-17A or IL-17F or B, C, D, and E? And then the other question, why does beta interferon exacerbate NMO? I'm not going to answer the second question. We have a very good handle on it, but it doesn't make for a highly entertaining story while dining unless you're a member of a small group. But anyway, on this 17A versus 17F, all of us make more 17F than 17A anyway in response to an infection. It's just, it's much more common. And I go and talk to a lot of colleagues, and they say, well, we saw a study where they didn't see anything interesting about 17. Most of the people will buy a kit, and most of the kits were made in response to to measure the first member of that family, 17A. So it turns out that it's inconvenient, but you have to have reagents and ways of studying not just one of the cytokines, but all the members of the family. And I say this knowing that I haven't studied B, C, D, and E because there aren't good reagents available, but there may be even more amazing stories that come out there. So there's a lot known. My colleague at Stanford, Chris Garcia, has taken X-ray crystallographic pictures of the complex of these 17A and 17F with their receptors. And it turns out that even though there's more 17F in your blood, 17A binds with about 100 times more strength to its receptors. So a lot of the biology has to be worked out. And Bob Axtell looked at the organization of the IL-17F and 17A genes, and F conveniently has elements in the gene that are sensitive to beta interferon. So that's why we're seeing a regulation focusing on 17F and not 17A. The important thing here is the science is clearly important and the mechanisms are important, but one of the take-home messages is going to be there could be a very effective therapy at hand to be tried experimentally to see if it can make a difference in NMO and IL-17 versions of MS. Sounds like obvious. Why aren't we doing that? If people have high IL-17 and you're telling me it's so bad, why don't we take an antibody to it? You know, I just came back from Oxford from a meeting organized by my colleague Mark Feldman, and in the 1980s he made the observation that of all the cytokines in the joints of people with rheumatoid arthritis, he felt from his experiments that TNF was the most important. So he tried an antibody to TNF, and people with rheumatoid arthritis, about 60% of them, got a lot better. And that was the early 90s. 20 years later, 2 million people in the world have taken anti-TNF, and it changed their lives dramatically and for the good, those people who were in the responder group. So this could be, if everything went according to what we hope, as important for people with
NMO and the IL-17 versions of demyelinating disease as anti-TNF was for people with RA. I sit in those meetings listening to the glories, by the way, of anti-TNF. It works brilliantly in diseases, other diseases like psoriasis and inflammatory bowel disease. But you know, the anti-TNFs carry a black box warning label because it makes demyelinating disease worse. And one of the other things that we found out from this research is that beta interferon, by the way, does not work in rheumatoid arthritis. And one of the things we found in our studies is that beta interferon actually drives TNF, so of course it wouldn't work in rheumatoid arthritis, and maybe that's why it does work in certain forms of demyelinating disease, whereas the antibody, the one that works so well in rheumatoid arthritis, carries a black box label. So all these intricacies, the puzzles are becoming more apparent. So again, I put here which would be a therapeutic target, IL-17A or F. Clearly right now I'm voting on 17F. Now, we can take this a little further. When I was at the meeting in Oxford, I was asked, hey, does beta interferon do anything in psoriasis? Because psoriasis now carries the stamp that it's an IL-17 disease. And I'll show you some papers that lupus and psoriasis are both examples of IL-17 diseases, and both of them get worse if you give beta interferon. So this was a dermatology review discussing the next topic that I'm going to get into, namely that you see a lot of neutrophils in the skin in this IL-17 disease, psoriasis. And then a couple of papers, IL-17A is increased in new onset lupus patients, and a paper from one of our neurology journals saying a case of psoriasis broke out in a person who had maybe the most subtle form of subclinical psoriasis, but as soon as they got beta interferon, boom, the psoriasis exploded. So back to the granulocyte story. Now we're going to focus on the issue about granulocytes in neuromyelitis optica. So Dr. Weinschenker and his colleagues, Claudia Lucanetti and the Mayo Clinic group, who deserve so much credit for their incredibly important research on defining the main target of the immune system in NMO, aquaporin 4. They've also looked carefully at the pathology. So they had a paper in the journal Brain in 2002, which showed that the granulocyte and the eosinophil were right at the scene of the crime in NMO. So that was the paper. When we looked at the our animal studies, the Th17 versus the Th1 form of EAE, from the back of the room it looks like paw prints in the Th17 as opposed to just dots in the Th1. Those paw prints are the multilobulated nuclei of the granulocyte. So you see it in EAE of the Th17 variety, and you see it in psoriasis, you see granulocytes, and in NMO, you see granulocytes. So this is the granulocyte. Finally, I get to show you a picture. It has a multi-lobe nucleus, so you're taking a slice through it, and you see 3 or 4 segments of the nucleus, and then it's filled with those granules that have all the explosive enzymes in it. They have names myeloperoxidase, they have names like defensin, and the one I'll talk about right now is called neutrophil elastase. So neutrophil elastase degrades tissue, it degrades extracellular matrix, and it's responsible for lung destruction and diseases ranging from COPD to cystic fibrosis. It's there in the granulocyte to fight infections. So we took a, oh my gosh, we're already at dessert, right? No, it's not about me, but I know there's going to be other speeches tonight, so we may truncate this talk halfway through, so because I know other people want to speak. So we took a drug that is in use in Japan for chronic obstructive pulmonary disease. And we asked if we took this drug that you can buy in Japan and tried it in our EAE model, would it work? So the drug is called Simvastatin. And we took a look at levels of elastase in plasma, and we find it's elevated. This was from the Stanford clinic run by Jeff Dunn. And then Bob Axtell did his experiment. The elastase inhibitor made the Th17 form of EAE better and had no effect, or made the Th1 form a little worse. So that could be another therapeutic, something from the family of elastase inhibitors, take a drug off the shelf that's approved somewhere and get it into a clinical trial in NMO. So the granulocyte is going to be quite important. Now, I'll just talk a little more about the proteomic studies that we're doing. This work was done by, again, funded by the Guthrie-Jackson Foundation. Mahon and Bill Robinson were the key people. This is a high-tech procedure where we actually look at the proteins either in lesions or in the blood. We've analyzed a couple of specimens really carefully. We have a very big catalog of what goes on in MS, so we can make a comparison. What are the differences between MS and NMO? So I put in bold, MS is dominated by lymphocytes, and a lot of the things we see in NMO is dominated by this neutrophil signature. Again, the neutrophil, so we see things like the elastase enzyme and the trypsins and antitrypsins. So we wanted to know, could we follow any of these markers in the blood, and maybe these markers could be a tip-off for somebody with NMO that they're going to have a relapse, and before the relapse happened, one might be able to use either a steroid and one of these elastase inhibitors and abort the damage, or anti-IL-17 and abort the damage. So in a collaboration with Ann Cross at Washington University, we started looking sequentially at people during relapse and remission, and it looks like the one of those neutrophil markers, the GRO-A, gets elevated. So these studies have to get revved up, but if you had to take a blood test on a weekly basis, or even have a finger stick test, if it could be reduced to a kit, you might be able to avert a flare-up of NMO in a very intelligent way, and then avert it with a medicine that would combat exactly what is going up. So the final thing, and I'll make it fast, is the last area that I think really deserves a shot, especially in NMO, where the Van de Lennen and colleagues have really put their finger on one of the most important immune responses. It's targeting aquaporin 4, and we've been, again, using some of these proteomic techniques to ask what portion of aquaporin 4 is being targeted, and this is a collaboration that's really been helped by the Guthrie-Jackson, not only in funding the research, but in making reagents available. So again, to make a long story short, we know some of the domains on the aquaporin 4 that are important, and I'm not going to go into it at this hour because I want to turn the podium over to others, but we've developed an experimental technique that is begging to be tried in myasthenia gravis and in NMO, whereby we can potentially, and we've done this in a few hundred patients now with MS and type 1 diabetes, if we know the antigen being attacked, we can shut down in a highly specific way the immune response to that antigen. So I'm not going to take you through all that right now, but that would be of the therapeutic options that I've raised tonight. We could inhibit some of the enzymes of granulocytes. We could target cytokines that are causing the trouble, the IL-17F, we could potentially shut down an immune response to aquaporin 4. So in the future, I could see that somebody has the diagnosis of NMO, and you do bring out the whole army and stop the disease initially with drugs like Rituxan. But after you get a ceasefire, you then want to be, if you can, much more specific about controlling the disease. And we're looking at, can you do some of these therapies even when your B cells have been taken down with Rituxan? So I think that there's going to be a lot of very promising ways of getting diseases like NMO into remission. There already are good ways of getting into remission, but getting continuing remissions without some of the heavy artillery. And I see in the future the intelligent use of blood tests to decide when to give a medicine and what medicine to give. And then finally, and it may take a while, to try to extinguish that immune response that drove the whole thing in the first place. And maybe in NMO, it's that immune response to aquaporin 4. So I'll just flip to the last slide. And no one asked me any questions, but I'm really happy to take your questions. Well, I remember in Ferris Bueller, who was the high school teacher who said, anyone? Anyone? I think it was Ben Stein. Yeah. Anyone? I guess everyone was in. All right. Well, Ben, I'll turn it over to you.